Hello, everyone. I'm Chunrong, Associate Professor of Environmental Health at the University of Memphis. This primer on air pollution exposure is collaborative work with Dr. Hector Alvarez at Oregon Health and Science University and Dr. Christina Fuller at Georgia State University. This is one of the primers that provide an overview of fundamental topics in socio-environmental research. By the end of this primer, you are expected to define air pollution and classify air pollutants, describe major health outcomes from air pollution exposure, describe measures of air pollution exposure, and discuss their accuracy, advantages, and disadvantages, and apply the knowledge in future air pollution studies from the public health perspective. In this primer, I will explain the basic definitions in air pollution, describe air pollution exposures, and the associated health effects, discuss different air pollution exposure measures, and briefly talk about major air pollution research themes in the public health domain. Air pollution is a broad scientific research area. In public health or environmental health, we only care about the air that the general population is exposed to. Thus, the air here means the air in the troposphere, typically in the range from the ground level to about 20 meters high. The air pollution concept is based on the concept of clean air. The clean air consists of nitrogen, oxygen, water vapor, carbon dioxide, inert gases, and trace level organic vapors. The clean air is a necessity of life and is considered harmless. Thus, air pollution is defined as the addition of harmful substances to the clean air or the elevation in concentrations of some air components, such as carbon dioxide and organic vapors. Air pollution can occur in any places, including indoor and outdoor environments. The term air pollution typically refers to ambient or outdoor air pollution. Air pollution that occurs in indoor environments is specifically called indoor air pollution. There are two major groups of air pollutants that have health concerns. Criteria air pollutants are a group of very common air pollutants regulated by Environmental Protection Agency on the basis of criteria. The term criteria means information on health and environmental effects of pollution. In the US, criteria air pollutants are regulated by National Ambient Air Quality Standards. Particulate matter, PM, nitrogen dioxide, and ozone are the three criteria pollutants that have received the most attention. It should be mentioned that ultrafine particles and black carbons are PM components. They have important health implications, but are not regulated by the national standard. Hazardous air pollutants or air toxics are those known to cause cancer and other serious health impacts. EPA regulates emissions of 187 air toxics, but not their concentrations in the ambient air. Air toxics include three major groups: volatile organic compounds, such as benzene, toluene, and formaldehyde; semi-volatile organic compounds, such as polycyclic aromatic hydrocarbons, PAHs; and heavy metals, such as mercury, cadmium, and chromium. Air pollutants can be categorized by source or by physical and chemical characteristics. An air pollutant may be directly emitted from a source, which means it's a primary pollutant, or formed in the atmosphere through physical and chemical conversion of precursors, which means it's a secondary pollutant. For example, carbon monoxide emitted from a car tailpipe is a primary pollutant. However, ozone, a secondary pollutant, is formed in the atmosphere when sunlight chemically converts other pollutants into ozone and other oxidant species. Another important feature of air pollution is whether the emissions are natural or from human activities. Anthropogenic sources are the major contributors to air pollution. Naturally occurring pollutants include volatile organic compounds from vegetation, pollens, volcanic gases, and dust from deserts. In physical form, air pollutants can be gases or particles. A pollutant's physical form and chemical composition affect the pollutant's ability to penetrate the respiratory system, as shown in the next slide. A person is exposed to air pollutants via inhalation, mainly. Sometimes the exposure can occur via dermal contact. For example, dermal exposure to trihalometane gases during the shower. A third way is ingestion. Large particles deposited on the upper airway may be swallowed to the stomach. Gases can directly reach deep into the lung. Particles of different sizes deposit in different regions of the respiratory system. Exposure to air pollution has been recognized as a major health risk factor. For example, the 2013 Global Burden of Disease study found air pollution is the number 5 risk contributor to global disability-adjusted life years, DALYs. Numerous studies have found air pollution exposure is associated with many health outcomes. For example, death, cardiovascular diseases, respiratory diseases, adverse birth outcomes, and cancers. The associations are reflected by increases in clinical visits, ER visits, school absenteeism, and hospitalizations. How do we measure air pollution exposure? If you have watched the exposure assessment primer, you should know there is a source to health continuum. Air pollution exposure can be measured at any stages of the exposure continuum, from the source to ambient concentration, microenvironmental concentration, personal concentration, and biomarker concentration. These measures have a gradient of accuracy, which with personal exposure and biomarkers as the best exposure measures. Next, we will briefly talk about each type of exposure measure. Biomonitoring refers to measurements of concentrations of biomarkers in human indicator media, such as blood, urine, feces, hair, or breast milk. Biomonitoring measures the total exposure, which is integrated over all sources and routes of exposure. If a chemical exists mainly in the air, then the biomarker can represent the exposure. However, if a chemical exists in the multiple environmental media, the focus on biomonitoring may be misleading. In such cases, environmental monitoring may be a more appropriate approach. Personal monitoring of air pollutants means collecting air samples from the air in an individual's breathing zone. The personal exposure measurements can best represent the actual exposure. However, personal monitoring is expensive and time-consuming and may interfere with a person's behaviors. It is well known that people spend more than 90% of their time in various indoor environments. Thus, concentrations of indoor air pollutants should better represent air pollution exposure than outdoor concentrations. Microenvironmental air monitoring means collecting air samples in different indoor environments, as well as outdoor air, for example, in homes, offices, schools, shops, and vehicles. The time-weighted integrated exposure is then calculated with microenvironmental concentrations and time spent in each microenvironment. In the US, ambient air monitoring is part of the regulatory environmental monitoring systems. It is the systematic, long-term assessment of pollutant levels by measuring the quantity and types of certain pollutants in the ambient air. There are several air monitoring networks in the US. The Air Quality System, AQS, is a national repository of ambient air monitoring data collected by EPA, state, local, and tribal air pollution control agencies. It also contains the meteorological data, descriptive information about each monitoring station, and the data quality assurance and quality control information. AQS data have been widely used for regulatory and research purposes. Monitoring data are not always available for everyone in the population of interest. Thus, a model can be used to estimate air pollution exposure for individuals or specific locations. As mentioned above, personal exposure can be modeled with microenvironmental concentrations and the time activity pattern. Ambient air concentrations are often modeled using dispersion and land use regression models. Air pollution exposure can also be represented by non-pollution type of surrogates. These surrogates could be air pollution emissions, for example, fuel consumption and air toxics emissions. They can also be activity patterns, such as distance and timing of travel or duration of work and its locations. Many exposure surrogates can be found from EPA's EJ Screen database, Smart Location database, and EnviroAtlas database. In the environmental health domain, information on air pollution exposure is often used in two types of research: environmental justice and environmental epidemiology. Numerous environmental justice studies have documented the clustering of emission sources and elevated air pollution in low-income and African-American concentrated neighborhoods. For example, I conducted a disparity research and found people in low-income areas have higher exposure to air toxics in the Cancer Alley in Louisiana. Another major application of air pollution exposure is environmental epidemiology. Numerous studies have investigated the association between air pollution and health outcomes in the real world, typically in large populations. Ambient air monitoring data are often used as surrogates for individual exposure. That is, one or more monitors placed in a city are assumed to represent citywide exposures. The potential weaknesses are the limited ability to control for confounders and the difficulty of accurately estimating personal exposure. In summary, air pollution exposure has been linked to many health outcomes. There are multiple ways to indicate human exposure to air pollution. Biomonitoring and personal monitoring are the direct measurement methods, and environmental monitoring provides the most available data. Modeling provides individual level data, and exposure surrogates are useful when monitoring data are missing. Air pollution exposure data are widely used in environmental justice and environmental epidemiology research. These are some references and readings relating to this primer. This work is supported by the JPB Environmental Health Fellowship Program. Thank you.